Hello, 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 everyone. Good morning, friends. Good morning, friends. Let's have a drink of our coffee or our tea together. Good morning. Good morning, friends. Okay, so today I want to deliver a new lecture on plasmid evolution because plasmids are very, very important in biotechnology, as we have discussed and as we have been learning. And it's important to understand the principles and the themes that guide plasmid evolution. So I thought this would be a good new lecture that we could do today. So one thing that's important to remember is that if this is a cell, let's call this E. coli, and E. coli has a chromosome, so green will equal chromosome. And there are plasmids in this cell. It's important to know that the evolution of plasmids will be different from the evolution of the chromosome. So because plasmids are selfish elements, evolution and adaptation in plasmids and the selective mechanisms that act on plasmids will be different from mechanisms that act on chromosomes. Okay? They're separate. They don't have the same motivations. They don't have the same goals. They don't have the same trajectories in evolution. They're different. It's important to remember that. That's the first point. So you should consider them separate, selfish elements. Okay, and let's briefly review. I spelled it wrong. I'll fix it. Let's briefly review the purpose of plasmids. Plasmids often encode versatility. We talked about this term versatility in the eukaryotes versus prokaryotes lecture. And I drew the picture of the Swiss Army knife, which has all the different functions. And typically when we think of versatility, typically we think of eukaryotes, right? That was what I described in that lecture. And I described how prokaryotes usually are not versatile. Typically prokaryotes are not versatile. They care about speed. But prokaryotes acquire versatility with plasmids, okay? Many of the genes that are what you might call optional, so optional genes, can be found on plasmids, okay? So it's important to remember that plasmids in prokaryotes are for versatility. Plasmids acquire versatility. Okay, so let's talk about factors that influence plasmid ecology. One, plasmid mobility. Okay, by this I mean movement. And by movement, I mean transfer, host to host. Okay, that's what I mean specifically by plasmid mobility. It's when one cell has a plasmid, okay, and another cell doesn't. And by some mechanism of mobility, this plasmid can transfer to a new cell. And now that cell becomes plus the plasmid. Okay, that's plasmid mobility. Plasmid mobility is an important factor that influences the ecology of plasmids. Okay, plasmid indispensability. This is kind of a long word. Indispensability. Another word for this would be it becomes essential. Okay, so some plasmids are optional and some plasmids become essential. And if the plasmid can become essential, it will ensure its replication. So if the plasmid can become essential, it will ensure its passage into daughter cells. Okay, so plasmids often use tricks. Plasmids use, in English, what you might say is tricks to make themselves essential to the host. Okay, and I have discussed some of these examples. An example of this, can you think of any? Can you write it in the chat? Could you write in the chat an example of a plasmid becoming essential? Yeah, that's a good answer. That's good. So I wasn't thinking of that, but that's a good answer. This is a good answer. Good answer. If the plasmid can acquire antibiotic, I think what they're trying to spell is resistance. A resistance cassette, then the plasmid can become essential, but only in media with the antibiotic. Okay, so that's not completely essential. There are better examples. Let me give a second example. The toxin antidote systems, right? I described in the previous lectures, the toxin antidote systems are systems where the plasmid will kill cells that don't inherit it, okay? And another word for this is addiction. TA systems, actually, toxin antidote systems addict the host cell to the plasmid. It can't get rid of it. If they lose the plasmid, they die, okay? So this is a good example of a plasmid using a trick to make itself essential to the host. Let me give a third example. Plasmids can often capture genes. Plasmids can capture chromosomal genes. Okay, so if you have, let's go back up to this. If you have the chromosome and a plasmid, and let's say there is a gene on the chromosome, and let's say that gene codes for, let's say it's something that makes histidine. It's a gene that makes histidine, okay? If the plasmid captures that gene, so if that gene somehow jumps onto the plasmid and it's no longer on the chromosome, okay? In this case, the cell must keep the plasmid or it loses its ability to make histidine, okay? This is an example. It could be any essential gene. It could be any essential gene X, okay? It could be a gene for synthesis of uracil. It could be a gene for synthesis of a tRNA. It could be a gene for some essential process in the cell, okay? So another trick to become essential is that it might capture a chromosomal gene that's essential, like biosynthesis genes, etc. Okay. So there are many tricks where a plasmid can become what we call indispensable or essential. Here's 3 examples. 3 factors that affect plasmid ecology. Plasmid stability, okay? Stability. And by this, what I mean specifically, if you have a mother cell, okay, that has plasmid, and let's say they replicate by binary fission, and here's the daughter cell. If the daughter cell doesn't inherit the plasmid, if the daughter cells do not inherit the plasmid at high %, then the plasmid is unstable. That's what it means, okay? So if the daughter cells do not inherit the plasmid at a high percentage, then the plasmid is unstable. Compare this to, versus a mother cell and a daughter cell where both cells inherit the plasmid. This would be a quote unquote stable plasmid, okay? And we're going to discuss today factors that are important for plasmid stability. Okay? 4, final factors that contribute to plasmid ecology are the contents of the plasmid. Or another way to say this is genes on the plasmid. Okay, and we discussed an example of this. If you have a plasmid that encodes conjugation genes, this plasmid will be more stable. It will be more stable. And so it will affect the ecology of that plasmid. So the gene contents of a plasmid will directly affect the inheritance and the mobility and the stability of plasmids. And this is one example. Example one. Let's give another example. Example two. Imagine you have gene X, any gene, and gene X gives a fitness benefit, gives a plus fitness benefit. Okay. This plasmid will be more stable because all the cells want to be a cell that has this plasmid if it increases fitness. So a gene that increases fitness it could be perhaps a metabolic operon, maybe the ERA operon. This is just an example. Okay. If the cells have an ERA operon on a plasmid, they will be able to digest arabinose, an extra sugar. Okay, so these cells would be more quote unquote versatile when compared to cells that don't have the ERA operon. Okay, so deletion, it's short for they don't have it. So these are some examples of how the genes on a plasmid can impact its mobility and its stability and its indispensability in a cell. Okay, good. Let's talk about processes that plasmids undergo. So let's talk about plasmid processes and evolutions. Okay. One, a term called invasion. Okay. So what we mean by the term invasion is imagine you have one cell that has a plasmid and you have a big population of bacteria that lack the plasmid. Okay. And let's say this is generation one. And let's say this is generation two. Maybe there's a way for me to do this faster. Okay. The process of invasion would be in generation two, it's in the population more. And in generation three, it's in the population. On and on and on and on until it's 100% in the population. This is what we call invasion. Plasmids undergo a process of invasion. Okay. So let's talk about mechanisms of plasmid mobility. Okay. So specific mechanisms that define plasmid mobility. A, conjugation. We already talked about that. Conjugation is when bacteria build little tubes and they can pass the plasmid bacteria to bacteria through these tubes. That's conjugation. We already discussed that. We remember that. And remember that conjugation machinery is often found on the plasmid because it makes the plasmid more stable. Natural transformation. Okay. So transformation is the process where bacteria picks up environmental DNA. So let me draw out what a natural transformation would be. Imagine you have one bacteria, okay, bacteria one, and it has plasmids, okay, and it might spit out, okay, when I say spit out, spit in English is like spitting out. Bacteria can spit out a plasmid into the environment, okay, into like the soil or wherever it lives, the environment, okay? And natural transformation is when a second bacteria, okay, sees sees the plasmid and it picks it up, just picks it up. That's natural transformation, okay? In some cells, this is high probability, and in some cells, this is low probability. There's a term called competence or natural competence. This is the ability of a bacteria to pick up environmental plasmids, okay? Or environmental DNA, nucleic acids. That's called natural competence. And natural transformation is when a plasmid goes through this process, okay? And a thing to think about is how stable is DNA in the environment? Well, remember that DNA is double-stranded, okay? DNA is always double-stranded, and that contributes to its stability, okay? So DNA is very stable. It's very stable, okay? In contrast to like RNA. So if you were to compare this to RNA, RNA is single-stranded, okay? And RNA is unstable. So RNA will get degraded really fast, like that, okay? But DNA is pretty stable. So this DNA in the environment is stable for long periods, okay? So you might imagine a scenario where a population of bacteria with a plasmid spit the plasmid out into the environment, okay? And maybe this population of bacteria dies. They die. And later a second population of bacteria invades this environment. This DNA will still be around. It will still persist. DNA can persist, okay? And so then these plasmids can be picked up later by natural transformation, okay? What number are we on? C. Virus mediated transfer, okay? So we're talking about mechanisms of mobility of plasmids, okay? So if you have a virus, let's say it's a bacteriophage, okay? And bacteriophages inject their DNA into bacteria, okay? And sometimes that DNA can integrate onto the chromosome. This is called lysogenic phages, okay? A bacteriophage is a virus that infects bacteria. And when we say phage, that is short for bacteriophage, okay? And some bacteriophages are lysogenic versus lytic, okay? So in lysogenic phases, the lysogenic phase is when the DNA gets integrated into the chromosome. And a lytic phase is when the virus particles are being made and they lyse the cell, okay? So there's two different life cycles of lysogenic phases, viruses. And sometimes this life cycle process of lysogenic phages can transfer plasmids via a few mechanisms. Okay, let me draw out some examples. Okay, imagine you have, let's do a bacterial cell, and here's the chromosome, and here's a plasmid. Okay, so this is a plasmid. Imagine a virus. Okay, let's get a different color. We'll get rainbow. A virus injects its DNA into the cell, and imagine the virus integrates onto the plasmid, okay? When viral particles are then assembled in the cell, they might pick up that plasmid, okay? And so you might have a virus particle that's perhaps carrying some chunks of DNA. And then when this exits the cell and invades a new cell, it might inject some of those sequences into the next cell, okay? So viruses might carry and transfer sequences of plasmids, okay? It might also, viruses might also enhance remember what I called spitting out of plasmid DNA. Imagine this cell has lots of plasmid in it, and imagine the virus goes into its lytic phase and it ruptures the cell, and spills a bunch of plasmid everywhere. In this way, viruses might also enhance the spitting out of plasmid DNA, spitting it out into the environment. Okay. Now let's think about these three mechanisms: conjugation, natural transformation, and virus-mediated transfer. Conjugation is active. By that, I mean like the purpose is to transfer DNA. The purpose is to transfer DNA. Okay. But natural transformation is passive. It's kind of an accident. It's an accident. Same thing with the virus-mediated transfer. It's passive. This is passive transfer. It's kind of an accident. It's accidental. Okay. Okay. So these are mechanisms, A, B, and C are mechanisms by which plasmids can transfer. But there are mechanisms also that can inhibit transfer. There are also mechanisms that inhibit plasmid transfer. Okay. And some of these you might call cellular immunity. Okay. So let's detail some of these. Restriction enzymes. Okay. So in a cell, if a plasmid comes in, this is what we call foreign DNA. Okay. Or you might call this other. It's other. It's not, this is a bad analogy, but it's not me. It's not me. It's something else. So the E. coli, right? Like the E. coli, it's got its own chromosome. And E. coli sees that plasmid and it recognizes it as foreign DNA. It's other. It's not me. Okay. And so sometimes it will attack the plasmid. Sometimes it will try to kill the plasmid because it's foreign DNA. Okay. So restriction enzymes, they are defense against parasitic plasmids and against viruses. Okay. So in the cell, if it's invaded, the E. coli might make a restriction enzyme, and a restriction enzyme is like a scissors, and it will snip it, cut it, destroy it. Okay. CRISPR-Cas9 systems. Okay. These are guided nucleases. And the Cas9 is a nuclease that's guided by an RNA. And if the RNA recognizes a plasmid, it will attack it, just like restriction enzymes. Okay. So CRISPR-Cas9 systems can also be defense against parasitic plasmids and viruses. And I think in that next half of the class, we will talk more about CRISPR-Cas9, but we'll save that discussion for later. 3, plasmid incompatibility. Okay. I discussed this in a previous lecture. So let me ask a question. What plasmid elements control plasmid compatibility and incompatibility? Write it in the chat. What plasmid elements control plasmid compatibility and incompatibility? Very good. Cheng Wu. Excellent. The origin. Excellent work. Great work. So if a plasmid jumps into a cell, and let's call this origin 1, and let's call this plasmid 2. So let's say this is one cell that has 2 plasmids, and they have the same origin. These plasmids will fight. These plasmids will compete because they are selfish. Okay. And they will fight until one wins. Okay. And by fighting, what I mean by fighting is if this cell divides, let's zoom out. If this cell divides by binary fission, This is the daughter cell. And let's say this plasmid gets inherited, but this one does not, then this plasmid wins. Okay, that's what I mean by quote unquote fighting. They're fighting for inheritance. Let me write that. They are fighting for inheritance and reproduction. Okay, so these are all factors: restriction enzymes, CRISPR-Cas9, plasmid incompatibility. These are all factors that inhibit plasmid transfer. Okay. All right, let's talk about how do plasmids evolve? Let's talk about that. Some really fascinating and interesting things can happen on plasmids. Okay. So on plasmids, how do plasmids evolve? Well, one, they can get SNPs. Okay. Every time they replicate their DNA, there is an error rate with the DNA polymerase. So a SNP, if you haven't seen it before, SNP equals single nucleotide polymorphism. It's just a mutation. It's just a mutation. Okay. So every time plasmids replicate, they can get a mutation. And then when they replicate again, they can get another mutation. Okay. And if there are 600 copies in a cell, and every division you get another 600 copies, that's a lot of mutations. That's a lot of mutations. Okay. So they generate a lot of genetic diversity just by single nucleotide polymorphism. 2. Indels. Okay. You'll see this. This is a nickname in English. Indels means insertions and deletions. You see this written in papers. Okay. So a plasmid, it could get something inserted. Okay. And that could yield plasmid that looks like this. Or you could have a plasmid, and let's say it's got a chunk. You can have a deletion, which yields a plasmid without that chunk. Okay. Those are insertions and deletions. We call those indels. That happens very commonly. Very commonly. 3. Coding. Let's call this coding versus non-coding mutations. Okay. So if a SNP, let's first do like a coding mutation. If a plasmid has a gene on it and there's a SNP in the gene, that would be a coding mutation. Okay. Versus, imagine you have a plasmid and you have a gene and you have the origin, and you get a mutation, a SNP in the origin. That would be a non-coding mutation. But this could affect copy number of the plasmid, and this could affect the gene content. Okay, so the different types of mutations can happen. And this mutation we would say it happened on the backbone. Okay, backbone. Again, in English, it's like your spine, your vertebrae, and we call this or like a frame, like the frame. Okay, so non-coding mutations, if they happen on the backbone, we call that the backbone. Okay, 4, bunch of things here: recombinations, translocations, and IS elements or transposons. Many of the insertions, so this relates to insertions and deletions. How do they mechanistically happen? They mechanistically happen through recombinations, translocations, and insertion sequence elements. Okay, so when you see IS, that equals insertion sequence element. That is the same thing as a transposon. So let's discuss this. If you have a plasmid and you have a chromosome, and this is inside of a cell, and let's say you have a gene, okay? Let's draw out each of these mechanisms. Mechanism 1, if the plasmid recombines with the chromosome, okay, that can yield a plasmid that looks like this, okay? That's a recombination. Okay, let's draw. This is also, you could also say that this is a translocation. Translocation. Something is moving from A to B, here to there. Let's discuss the transposon. Okay, so imagine you have the cell, chromosome, and a plasmid. And imagine you have gene X, and a transposon. Call that T. T equals a transposon. Transposon will make messenger RNA that codes for the T, and the ribosome will make a protein that's a transposase. Okay. And transposases bind DNA at recognition sites, and they grab their gene and they move it, and they can paste it. Okay. So transposons cut and paste DNA. And sometimes they make mistakes. Okay. So this might happen. Or sometimes they make a mistake and they might take a little extra and they might move it. Okay. So sometimes transposons make mistakes and carry other genes. Okay. So these are mechanisms of inserting onto plasmids, how they evolve, mechanisms of their evolution. Okay. Now let's talk about a very specific example of this called gene amplification. Gene amplification. Okay. Gene amplification is when you have one copy and then you somehow generate more copies of gene X. That's gene amplification. And plasmids can be substrates for gene amplification. Plasmids can be substrates or structures on which gene amplification occurs. Okay, let me show you an example. Say you have a cell, chromosome, gene X, and a plasmid. Let's get a different color, purple. A purple plasmid. And let's say the purple plasmid has an origin that's PBR322, which is high copy, 600 copies. Okay. If you have a translocation event where that plasmid picks up X, okay, how many copies are you going to have after that translocation? If you translocate a gene X onto a plasmid with PBR322 origin, now after, how many copies of gene X do you have? Write it in the chat. If you translocate gene X onto a plasmid with PBR322 origin, after the translocation, how many copies of gene X do you have? It's not a trick. Yes, good. It's not a trick question. Very good. Zini Dong, very good. You're gonna have 600 copies. Okay, so you can a translocation, one translocation event of one gene with one copy on the chromosome can lead to 600 times copy number. Okay, does this make sense? That's gene amplification. That is gene amplification. Okay, that can have huge effects. Okay, that can have massive effects on a cell. Imagine, what would be a good example? Imagine this was a gene for detoxification. If this was a gene, if gene X was a gene for detoxification, this cell might now be super powerful at detoxification. Okay, so this can create situations where the bacteria evolves. Let me give you a specific example of where this happens. Okay, example one. Okay, there is a pathogen of Lyme's disease. Okay, Lyme's disease is spread by ticks. Okay, so let me get a picture. I'll show you. You'll recognize them. Hang on. Ticks. You recognize this. You know this. Okay. Lyme's disease. The pathogen of Lyme's disease is called the genus name is Borrelia burgdorferi. Okay. And ticks carry this bacteria. It's bacteria called Borrelia burgdorferi. Okay. And Borrelia burgdorferi has many tiny little chromosomes, almost like plasmids. Okay. And on those little nucleic sequences, it has many, many, many copies of one gene. Okay. And the one gene that it has many, many, many copies of is an outer surface protein. Okay. So in Borrelia burgdorferi, the outer surface protein is gene amplified. They have many, many, many copies of it. And then outer surface protein, what is it? Okay, let's draw what an outer surface protein is. Okay, if you have a bacteria, there's an outer membrane, and outer surface proteins go into the outer membrane. So an OSP, outer surface protein, they go into the membrane. Okay. So in Borrelia, there's many, many, many of these outer surface proteins. And what happens is they evolve. Okay. They get mutations and they change and they recombine with each other. So let me ask a question. Why would Borrelia burgdorferi, which is a pathogen, why would it want many, many copies of outer surface proteins? This is a hard question. I'm testing your knowledge. Think about it and write it in the chat. Why would Borrelia burgdorferi want many, many, many copies of outer surface proteins? And think about this. Think about the fact that it's a pathogen. Why would Borrelia burgdorferi want many, many copies of outer surface proteins? I'm going to wait. I'm going to wait until you answer it. Until you take a guess. I'm just going to wait here. Yes. Very good. Very good. Cheng Wu. Very good. Excellent. Excellent work. Very good. So Borrelia burgdorferi is a pathogen. Okay. And in the bloodstream, humans make antibodies. Remember the antibody discussion in the data structures? Humans will make antibodies, okay, that recognize antibodies, that recognize the outer surface proteins, okay? And so if Borrelia is recognized by antibodies, it will die. It will be killed by the immunity pathway, okay? But if it can change its outer surface proteins, if it can quickly evolve its outer surface proteins, it can hide from the immune system, okay? Very good. Excellent work, Cheng Wu. So now you see why bacteria might want to amplify certain genes. You see why this might be useful. So plasmids can promote gene amplification, and this is one specific example of why this could be very, very useful. Okay. Okay. Let's talk about how else other ways that plasmids evolve. Okay. So I think this is 3 or C, I can't remember. Host range. What we mean by this is how many different species of bacteria can carry and replicate the plasmid. That's what we mean by host range. Host range means this. How many different species of the bacteria can carry and replicate the plasmid? Well, we already know what's important. One of the things that's important is the origin, okay? So one thing you might see plasmids do is plasmids might pick up a few different origins. Origin 1, origin 2, origin 3. And this might be useful. So carrying extra origins might be useful for the plasmid because it will expand the host range. Okay. So imagine in E. coli, we have one plasmid, and let's give it two origins. Let's give it origin 1, origin 2. Okay. And in E. coli, the origin 1 is important. But let's say, imagine in Bacillus subtilis, and we will talk about Bacillus later in later lectures. Imagine in Bacillus, origin 2 is important. Okay. This plasmid can replicate in both species of bacteria because it has multiple origins. Okay. So plasmids might want to carry extra origins to expand their host range. Let's also talk about this protein called DNA A. Okay. DNA A is a gene that codes for messenger RNA, that's DNA A, which codes for a protein, DNA A. Okay. And DNA A is the actual protein that binds the origin. Okay. So I think in a previous lecture, I drew the origin and I drew it binding DNA polymerase. Okay. That was too simple. This is too simple. I lied. Okay. What actually happens is DNA A mediates the interaction for DNA polymerase. Okay. So DNA A is a very, very, very important protein. Okay. This is the truth. This one. Okay. The other one is a little bit too simple. But some plasmids, now let me relate this to plasmids. Some plasmids, they can evolve their own DNA A gene to specifically recognize their own origin of replication. Okay. So if, let's say they get a DNA A gene, and let's say it recognizes its own origin, how will this affect host range? How will this impact, or how will this change plasmid host range? This is another hard question. Think about this and give me an answer in the chat. If the plasmid, I'll write out the question specifically. If the plasmid acquires its own DNA A gene, how will that change host plasmid range? Write it in the chat. And I'm just going to sit here until you answer it, drinking my coffee. Think it through. You can do it. You guys said you were the best. If you're really the best, you can answer this question. Okay, let me help. Remember, DNA A binds origins and recruits DNA Pol. And think about host range will be influenced by DNA A's ability to bind origins. Okay. Think about this. You can do it. I don't want to give you the answer yet. If a plasmid acquires its own DNA A gene, which recognizes its own origin, how will this affect host range? You have two options for an answer. Okay. You have two options: expand or shrink. Shrink in English means go down. Expand means go up. Which one? Write it in the chat right now. No more waiting. Nobody wants to be first. Is it really that hard? Write it in the chat. Come on. Don't be afraid. Have courage. There's two options. The longer you wait, the longer I'm going to wait to give you a break. Come on, just pick one. Is the chat not working? Thank you, Cheng Wu. Thank you. Good. Ching Wu got it first, but these are some of the others. Good. Expand. It will expand. Let me explain why. Okay. Let me explain why. Why is this correct? It's correct because the origin no longer needs the host's DNA to bind, to replicate. Okay. So the plasmid is now independent of any specific DNA interaction. This plasmid could, in theory, replicate in any bacteria. In theory. Now, it won't work like that. But this plasmid, which has its own DNA, this one, that could, in theory, replicate in any bacteria. Okay. Because it has its own DNA, which recognizes it, and it's going to recruit the DNA polymerase because the DNA polymerase is very conserved. Okay, so I guess the key to understanding this problem is DNA polymerase is conserved. It very rarely mutates. Okay, so the interaction site between DNA A and DNA Pol is probably conserved. Okay. But in contrast, the interaction site between DNA A and the origin is quickly evolving to be host specific. It's probably quickly evolving all the time. Okay. So this is the key to understanding why that answer is correct. Okay. And if I was to draw this out, if you have the origin and you have DNA A and you have DNA Pol, okay, this one is changing fast, this interaction, and this interaction is changing slow, okay? So if you get your own DNA A, this is never going to change. So you'll be able to, in theory, replicate in any bacteria, in many bacteria. Okay. So plasmids might acquire their own DNA A protein or gene. Let me also discuss another gene, which is called PAR A and PAR B. So there's different genes, PAR A and PAR B. Okay, you often find PAR A and PAR B on plasmids. So you often find on plasmids. And PAR A and PAR B are what we call partition proteins, okay? And what they do is you have a cell and a daughter cell, but let's draw them not yet cytokinesis. And you have plasmids, okay? You want the plasmids to be distributed evenly. So if you have 1, 2, 3, 4, 5, 6, 7, 8, you want 1, 2, 3, 4, 5, 6, 7, 8. This is what you want. This is good. Okay. This is good for the plasmid. This is bad, right? Because this is going to be unstable. This will be stable. Okay. And so PAR proteins, PAR A and B proteins do this. They do this. So they're proteins and they mediate essentially like the partitioning of plasmids so that it's equal. So PAR proteins, zoom in. PAR proteins mediate equal distribution of plasmids. Okay. So these are all factors. These are all factors that are important for host range and division in plasmids. Let me talk about one final thing, which is loss of independence. So plasmid loss of independence. Independence means like you are your own entity. Okay. So Plasmids can evolve. Let me write this. Plasmids can evolve into chromosomes. Plasmids can evolve into chromosomes. Let me demonstrate how this might happen. Okay, so if you have a plasmid, the first step, step one, is become essential. Become essential. So it might acquire an essential gene, maybe like that hiss example. Okay, so become essential. Now you cannot be lost. The cell cannot lose the plasmid. Okay, at this point, after this, the fate, or you might call this the destiny. I'm trying to think of the best translation. The fate of the plasmid is linked to the fate of the cell. Okay. So now their interests are the same. At that point, now their interests are the same. The plasmid wants to survive, and it wants the cell host to survive. Okay. At this point, it can become a chromosome. At this point, step two, it can evolve into a chromosome. Or alternatively, or step three, it could fuse with the chromosome. Okay, so if there's a chromosome, the plasmid might, at this point, it might fuse back onto the chromosome. So there could be a couple things that could happen. But the point is that plasmids can evolve into chromosomes. Okay. All right. This is a large discussion on the evolution of plasmids.
Investigate real time to die. I show me death. I show me